Which of the following are correct about EcoR1? Cut the DNA with blunt end. No, this is wrong. So wherever A is there, strike it out. 2 and 3 has A. Next one. So if you notice B, C, E, B, C, D. So B and C are common and they are correct. So we have to rule out between E and D. Now D is such a long statement. First focus on E. Exonucleus. EcoR1 is endonucleus. So exonucleus will be wrong. E is wrong. So the answer is option number 4. So this is confirmed answer. Mark it on the OMR sheet. So we attempted this question in around, let's say, 50 seconds. So see, if you use this approach, you arrive at the answer, and I didn't even go through B, C, D. And I'm sure that this is the right answer. So use this approach of elimination to arrive at the right answer. So let's understand what is B, C, and D. B says EcoR1 cut the DNA with sticky end. Yes, correct. So the sequence it recognizes is GAA TTC, and the complementary sequence will be CTTAAG. So it will cleave the bond. It is endonuclease. So within the sequence it will attack. So it will break the bond here and over here. So after digestion, it will look something like this. This will be G, and this strand will open up. Okay. And on the complementary strand, you will have C, T, T, A, and A. This G will also open up. So this end is called, this overhang is called sticky end. So this statement is correct. Next one, recognize a specific palindromic sequence. So this is the palindromic sequence. So from this end, it will be 5 prime and this will be 3 prime. And when you read it from here, it will be GAATTC. Okay. So this is the reason why it is called palindrome. So from here or here, it will be read in the same way. GAATTC. Then last one is cut the DNA between base G and A. Yes, this is what I have shown here. When encounters the DNA sequence GAATTC. So this makes B and C and D, 3 statements correct in this question. So initially, both the source and the vector DNA will have the recognition sequence for EcoR1. C, T, T, A, and G. So this sequence is on, this is vector DNA. And then with a different color, I'm going to show you the source DNA. It will also have the same sequence. So now after digestion with EcoR1, EcoR1 will attack and break the bond, the phosphoester bond between G and A, and over here also. Here and here. So see, both vector DNA and source DNA are to be treated with EcoR1. Okay, and then sticky ends will be produced. So G, and this will be C, T, T, A. And source DNA after digestion will look something like this. Now see, this is AATT and this is TTAA. They complement each other and they will immediately pair up. And this is the reason why it is called sticky ends. The two will pair up and it will look something like this. So this is the fusion DNA. It has component of vector DNA as well as source DNA. A, the final ligation, because this bond is still broken between G and A. So that will be sealed by DNA ligase. So this is how cloning is done and recombinant DNA is produced. So practice this diagram. Okay, that will help you in better understanding and you will remember the details well. You will at least never ever forget the palindromic sequence of EcoR1. It is fun to draw these diagrams. So that's all about this question. Thank you so much for watching my video.